And there we go.
All right, I'm watching Instagram.
There we go. Let's see, do we like the... There we go. Don't see anybody yet.
We're going.
Awesome. Let's see, do you like the orange waves or do you like the like shiny little helix thing? You can't see it as well.
Yeah.
It's behind our faces.
Either way.
Fair enough. Also, you can't go wrong with oh, hey, we got some people coming on. Hey, guys.
Hey.
I like this background. It has Evil on it. We love Evil. She's a good girl. Hi, Zoda.
She could lay some eggs.
I would love Evil eggs. Maybe her kids will be as spicy as she is.
She's chilled out some over time.
That's good. Hey, Matt. And Sweet Slithering Delights. You were there with us on... Actually, I think all three of you guys were there with us on Tuesday. Good to see you again.
And we've got a few on Instagram, too.
Awesome. What's up, guys?
Garrick 489, Reptile Pueblo.
Yeah, they were there on Tuesday, too.
What's up?
Bald head balls.
For the...
Hi,
yeah. Oh, there's a delay for the Instagram one. I was like, oh. But yeah, it's the same. It's the same recording, but it's just delayed a little. Yeah, there we go.
Gotcha. Okay.
VA Morphs. Hey, Tyler. Celtic Reptiles.
Mm-hmm.
Dragon Soul Reptiles. Yeah, we got quite a few coming in.
Awesome. Yeah. Oh, well, I didn't even actually, be helpful if I actually did present. There, now we look official. Livestream Thursdays.
All right.
If you help us, if you have a better idea for what these streams end up being, a better name for it, let us know. I think our vibe that we're going for is I don't know, we're already pretty casual for the Reptile Genetics Weekly, but this is just kind of a sit down, nerd out, maybe read out some live tests if people have fast test results.
Talk about cool stuff that's come out this last week. Cool YouTube videos people have put up, things like that.
Mm-hmm. Smash that like. Yes, please.
Yes.
Hi, Aaron. You've done 3 tests. Only 1 was positive, but still cool. Hey, at least you know.
Yes, and 1 is better than none, but hopefully
That's
that
true.
means your next 3 you'll get 2 out of 3 maybe.
Yeah. I think a while back we brought somebody on on the main show, and we had I think they got 5 no's and 1 het. I'm pretty sure. It was rough.
Oh yeah. I had someone, I should be able to remember who it was, but someone had Ultramel results. There were 5 of them that were POS-het Ultramel, and they got 5 out of 5.
5 out. Oh, that's nice.
Yeah.
Well, 5 out of 5, all of them positive.
All of them were het for Ultramel. I think they were 66% POS-het. So
that's
there's
good.
Some crazy good results sometimes.
Awesome. Love to hear it.
Jay Cox Reptiles on Instagram says that we sent some great results Tuesday night. So yes.
All right.
You have to remind me, Jay Cox, what we sent you, if there's anything you want us to shout out on here that you scored on.
Oh yeah, for sure. But yeah, I want to talk about Ben. You want to talk about some of the stuff you sent that you've been finding on Instagram?
Yeah, let's do that. One other question really quick from Instagram.
Oh, yeah.
Python Portal says, are you getting any sheds for Joliffe? Not many at all. I think less than 5, maybe even less than 3. So if somebody sent me several, there's 2 different addresses. The address that you send to for paid tests is in Texas, but a lot of the time people will put their ones, they'll say, hey, here's some lace or some mandarin or whatever for research sheds, and they'll put them with their paid tests that go to Texas, and then they'll collect them for a month or 2, and then they'll send them to me. So if yours went to Texas, that could be that they're delayed a little bit and I don't have them yet, but anyway, I haven't gotten them to me here in Virginia yet. So if somebody's sending them out, holler out, let us know to keep an eye out for them. But otherwise, we need lots more.
Yes. Hi, Blackrock Balls. What's up, John? Sota Balls says, or Soda. Oh, because Minnesota. I like that's cute. That's 4 out of 4 POS heads negative. No, I'm so sorry. That's rough.
A bummer when that happens.
Yeah,
we've had a couple larger breeders that were doing stuff with the bulk ordering, and they had males they had bred, but they didn't test the male. They just tested the offspring, and the male was a posset. So I'd feel bad. There'd be like 8 results, and they'd all be negative. I'm like, I'm so sorry. I don't know how you missed. They're like, oh, it's okay. The male was a posset, and I just ran the whole clutch for other things. So I threw, posset clown. I threw the clown test. So I always feel so bad. I'm like, there's 8 out of 8 are negative. Like, oh, it's okay. The sire was a posset. So
okay, fair enough.
Yeah.
So I mean, granted, that would be crazy odds if he was actually ahead, but just kept throwing negatives. That's my, like, who isn't at work right now? I am and I'm not.
Yeah. Very, very legit, Sloan. This was just the time we could do it. So we figured we'd get on here and nerd out for a bit. And if more people can't watch it till later, they can watch the replay later. So
yeah, exactly.
But so far, there's more people on than I thought there would be. We've got about 20 in YouTube and Facebook. And let's see. I'm not seeing the numbers on Instagram, but yeah, I see several with questions coming through. So yeah.
Awesome.
Thankful that, and if y'all are at work, then hopefully you don't get busted.
Lunch break. Breaks. Good for your mental health. Good for your productivity.
You are at work?
Well. Yeah. Cool.
Don't get busted.
Don't get busted. Yeah.
So yeah, some of the cool stuff we saw come through. Those of you that follow us on in social media will see some of these stories we put up. We got a really cool it's a paradox. It's an ultra male paradox, but it's just like a little bit of the face.
Yeah.
That was pretty cool.
I like that. I love that little face.
Yes. And
cool.
let's see.
Balls
and
of
then
Fury.
we had Balls of Fury. We had a cool Morph Monday, and that came from let's see, it says there, can you see who the original producer of it? There's a Florida breeders.
Yeah, Kittinger Reptiles.
Yeah. So beautiful job, Kittingers.
Mm-hmm. Someone's pulling eggs.
Yeah, we started putting some posts up highlighting people that are either getting eggs or ovs or post-ov sheds or eggs starting to hatch and first sheds from babies. So it's starting to warm up into the fun part of the year, that's for sure.
So exciting. And you're working from the comfort of your home, so no one to bust you at work here. Good, Aaron. I don't know why I say Marin.
Yes.
Do we have a lace test?
No lace test yet. We have more data rolling in here very soon. So cross your fingers for us. Maybe we'll get it.
Crossing everything I got.
Yes. And then I think it was Rice Reptiles that posted that last one.
Mm-hmm.
Yeah, that's a really cool one that is available. So jump on what you can.
Oh, he's pretty. I love that busy pattern. It's Hurricane that does that, right?
I think
at least in this combo.
Yeah, he had several. Yeah, that really busy pattern.
Mm-hmm. So
and then
cool.
The leopard and lots of good stuff.
Yeah. Let's see. These should be a little easier to read. I started doing 3s in these slides instead of 4s. That auction win though. I guess Balls of Fury won that. I don't know who produced it, but it's pretty.
Yeah. I think it was Python Shed. But yeah, that was an awesome score. I bet that one wasn't. I would imagine as an auction got a good deal, but probably not the cheap ones.
Yeah, for sure. Valuable super vanilla. Yes. Oh, we've been needing super vanillas.
Yes.
That's what's up. Don't super vanillas usually come with like that little like blotch of white right on the forehead? Or am I thinking?
Kind of whites out their head. Yep, you're right.
That's cool.
Yeah. and that comes from a local person to us. That's from Dustin. And I
That's
think he
right.
said that she's been locking up and growing follicles, so you might have some babies from her. So,
Atta girl. That's awesome.
Yes.
Ooh, banana test soon from Matt Krigo.
Yeah, that's another one we're working on and getting stuff coming through. So we don't have it yet, but we're working on it.
Yeah. From Sweet Slither. I think we both clicked on that one, Ben. Sweet Slithering Delights, Vanilla Mahogany, and Tri-Stripe test. Yeah, a lot of people have been requesting those.
Yeah, all of those are in process, so they're coming.
Yay!
So awesome. The writing's too small for me. What's that last one? What all's in it?
Yeah,
that's from Bomb City.
Wookiee, Spot Nose, Black Pastel, GHI, Mojave, Yellow Belly, Het Clown. What a beast!
Yeah, all kinds of good stuff in that one. That's pretty cool.
Nice. Really such like a there's not a lot pattern wise going on, and then you read the label and you're like, whoa, it's amazing.
Oh yeah,
pretty. I love the color. Yeah,
Balls of Fury says on Instagram it was Josh Jensen. That's right.
Josh Jensen.
Jay Royals Reptiles was the one that produced that. That a really cool. There you go.
Nice. That's
Thanks,
awesome.
Mike, if that's you, thank you.
Hi, Mike.
And Python Portal says they'll be sending in Joliffe stuff. So yeah, we just don't really get very many questions about Joliffe or anything. So yeah, send them in.
Yeah. Let's see, from Soda Balls. What is the reality of the Black Rider genes, Red Gene, and Ringer Gene? I have.
Yeah, great question. I don't know for sure if we will be able to tease out the genetics behind those. I don't know for sure. Those are ones that will probably be even more complicated than we talked a little bit about on Tuesday about how high OD and the extra dark axanthics, how those projects are going to take a little longer because they're not there's the primary mutation that makes it an OD, or the primary mutation that makes it axanthic, and then we're assuming there's a secondary mutation that then will cause it to be either extra dark axanthic or high OD. So finding those secondary mutations will be a little tricky. So something like the red gene and ringer gene, those will probably be pretty difficult to tease out. But you never know. If we have enough people purchasing the tests we have available, and I can keep playing, we have enough to pay for people to be searching that out, me and other people as well in the future, keep working on it, then eventually hopefully we can get there.
Yeah. Question from Roxanne. Are there two different lines of blackhead? One seems to be brighter than the other, like black pastels, or are they just variations?
We haven't seen that in testing yet, but as you all that watch the show know, sometimes like with Ultramel, we have the Ultramel A is more common, but I would say about a fourth of the time to a third of the time we'll see Ultramel B. But on the other hand, with Yellow Belly, the -1 we see almost all the time. The -17 we only see like one out of I would say 300. So if there are really two different mutations that cause blackhead, if it's 50-50, we should have caught it already. If it's 1 in 300, we might not have seen it yet. So if you have any, Roxanne, that you feel really confident, this one is one, this one is the other, I would love to just send them in. If you go to our website and go to the Help Us and just say, hey, on the live, we asked about potentially two different lines of blackhead. Here's two really good, differences, and send us pictures to our Instagram or Facebook messaging, and we can check it out.
Cool beans. Let's see. Anything new for NR Mandarin test? How do you pronounce NR?
I think it's just NR. Maybe
Okay.
someone here can give us more. I don't know the background on that. A lot of
Yeah.
people say Mandarin, and a lot of people say NR Mandarin. I don't know
Hmm.
what NR stands for, so
Okay.
I'm kind of older school ball python breeder. I've kept up with most of the newer things, but when I first was buying ball pythons, there was really only albino, piebald, pastel, spider, pinstripe. I mean, that was about all there was. Pinstripes at that time were like 25 grand a piece. Oh, and
Woo.
there were some fires back then. Um, I remember when Yellow Belly popped up, and lots of these others. So
Hmm.
yeah, if someone knows more about the NR thing, educate us.
Yes,
But
please.
but yeah, we do have quite a few sheds for Mandarin, and they're being sequenced. That's one we kind of talked about on Tuesday. Will likely be a little bit harder to tease out
Hmm.
because there's a decent chance some of them that have been sent in may think that they're homozygous, but actually het, or maybe actually het, or anyway, they're a little bit more difficult than like clown versus not clown to be able to know Mandarin versus not, or super Mandarin versus Mandarin. And anytime we tell the software the incorrect thing, it takes it a little longer for us to be able to find the actual mutation. And so things that are harder to make that call visually also make it harder to find them using the software. So
Yeah.
we're working on it.
Yeah. Oh, the part about Dalmatian pieds, I didn't know what that was, so I looked it up and I'm in love.
Yeah.
Look. Oh, that's so cool.
Yeah. I have one
Dude.
that got a little bit, but it was only like, 3 or 4. One of them came
Mhm.
out, it's like an X. It's pretty cool.
Oh,
But yeah, I haven't ever had one pepper up that much. That's pretty cool.
oh, that's amazing. Yeah, I don't this is just something I spotted on Reddit. I'm not sure what like like any lineage info on that, but that's so cool. I love that.
Yeah.
Um yeah.
Yeah. And as far as if people have those, send in sheds. If we have, we obviously already got a whole bunch of normal pieds. So
Oh yeah.
there's enough of those Dalmatian looking ones, and people send them in. We'll happily look them up.
Oh yeah. Roxanne, awesome. Thank you. Only one at the moment, looking to get another in the future. Regarding blackheads. Okay, cool. Oh, I think
We have a question on Instagram. This is from Duskfire Geckos. Are the genes that influence ball python phenotypes similar in other reptiles besides just name? Like, is clown or mandarin in balls the same allelic location and mutation as geckos, for example?
good question.
So, yeah. So, what we have seen not only within reptiles, but especially with like the albinos and the axanthics and hypos. So we see that kind of a phenotype or that kind of a look in mice, rats, zebrafish, angelfish, all kinds of different vertebrates, sometimes axolotl, some of the other amphibians, frogs, and stuff like that. So there really are like 4 or 5 main genes that when they have a mutation that's a very disruptive mutation that really messes up that protein, it can be a complete stop, like put a stop codon in there. It can be like a frame shift, something that really interrupts that gene. And it's in these 4 or 5. ACA2 is one we talked about. That's lavender
Mhm.
albino and ball pythons. There's, like I said, 4 or 5 that if you have that major disruption in humans too, then you can get albino. And so that I would assume would also be the same in geckos, would be a very safe assumption. But not all. Like, we don't know for sure what causes banana in ball pythons. It's a very similar type of look as an amelanistic or albino, some type of albinism. We haven't been able to find monarch, so there might still be some other genes that are different, but there certainly will be some, like the genes that cause albino in the geckos. I would imagine the majority of them will be ones we also see mice and rats and ball pythons and fish and all that stuff.
Yeah.
And specifically the one that does clown in ball pythons, we did a episode on that, and that's the same gene that causes humans to have red hair. And so yeah, that's pretty cool. So it's another one of those where different vertebrates, cattle, goats, mink, humans, snakes, rats, all of them, it tends to add more red or yellow coloration when there's mutations in that MC1R, I think is what it is.
I think so, yeah. Too cool. I still just can't get over. I love the like clowns and redheads are kind of the same mutation wise. That makes me happy.
Yes.
Yeah.
And Clough Family Reptile says Brian Bodie has a Dalmatian pied. He's trying to prove genetics. So yeah, I remember. I think I usually see him at least at the California show, the Pomona show that I've been going to the last couple of years, and looks like I'll be going to Pomona again. So any of you that are in California, the show that I haven't been to for a while that I plan to this year, I should be at Daytona this time. So.
Nice.
I think the last time I went was 2019, but yeah, I'll have to remember, if you all talk to Brian, tell him to send us sheds from those Dalmatian pieds, and when I see him, I'll bug him about them too.
All right. Mike's asking if we're going to Tinley.
So this weekend, no, but October, yes. So yeah, I will at least be there in October, and I think Sean will as well. And we'll have to talk about it. I think we've talked about it briefly in the past for Kayla. I would imagine you need to go at least once at some point.
Yes, I've never been to any Tinleys, so I really want to go. Plus, I know a lot of people there. I want to meet Ed and Emily of Snake Discovery too. They were kind of part of my introduction to the snake world too.
Oh yeah.
Most of it was like catching snakes, but then they kind of influenced it, especially the hog noses.
Oh yeah.
So
they didn't make it to the Texas show you went to?
No, they didn't, sadly.
Oh, that's bad. I think that's one they've done in the past, at least some, but
I think so. But yeah, no, they're so of course I want to see them, and I know Scott Hitchcock will be there, Clint Bartley will be there, and then a bazillion ball python people that I also know and love. So it'll be cool.
Yes.
Let's see. Tinley's awesome. When you went, only 2 vendors had TSK. And that's why you were there. No. That's okay.
I missed it. Vendors, yeah, okay.
Yeah.
Cool. Cold-Blooded Earth on Instagram is asking, can we send in Gila sheds for sexing? Yes. I do have a few, and I have done some sequencing, but I could definitely use some more. So whether it's Gilas or various species of monitor lizards or blue-tongue skinks, Egernia, any of those are ones that I would love to have more known sex sheds. So there's no reason to send in ones that you're not sure about yet for any of those. But yes, we would love to get more known sex sheds from all of those. Those are sex determination tests we're still working on.
That'll be cool when we have those. When I'm like at shows, like local shows, talking to people who keep Gilas, they're always like, wait, you could do that, like sex determination? That would be amazing. And they're like, yeah, do you have a card? Like, hook me up. Yeah, I know that's definitely like something people really want. So that would be cool.
Yeah, I know people have done it by ultrasound. Once the animals are large enough, they would be, sexually mature enough for the females to develop follicles. But being able to do it from that first shed, or even taking some of the membrane from the egg, that would be way better.
Fair, yeah. Let's see. How close do we think we're getting to an RK asphalt test? I know that's been one we've been hunting for as well.
Yeah, so with that one, we know we're pretty confident we know where to look. It should be in the same gene as all the other yellow belly complex. Excuse me.
You're good.
So yeah, we know what gene to look in. it'll just take a little luck of being able to hit it. So some genes are only a few hundred A's, T's, C's, and G's, but some genes are spread out over a long way, and they have many different sections to the gene that are, the functional part of the gene, but they're also spread out and separated by parts of the sequence that are called introns or non-coding. So some genes are harder to sequence than others, and we just haven't found. We do have enough. Well, we do have a few RK asphalt samples, enough that we should be able to find it from them. But any of you that have RK asphalt that is RK asphalt, or someone that does and haven't sent them in yet, definitely send them in, and having a few more would help us out.
Yeah. Also, I'm doing like a shameless plug of our we're just paying the bills with the little like all axanthic thing. If you're super into like axanthics, this is a special that we have going right now for the rest of March. So
yes,
get yourself in on that.
Jump on it while you can. I know it's already helped quite a few people. To be able to figure out what line of axanthic they've got. So.
But back to more questions. Mike was asking. Well, actually, that's kind of isn't a bad idea. Doing a promotion at shows, drop off sheds, and get a discount. Yeah. We've definitely partnered with shows before, like we did with Colubrid Fest, and put my face here while I'm talking. And could drive more traffic. I didn't think about it, maybe driving more traffic to the show. That would be cool.
Yeah, we'll have to see what we can do. Unfortunately, for a couple of people that brought paid tests to shows, and I didn't realize they were paid tests. I thought everything was research sheds.
Oh no.
So like I didn't look at them for a week or whatever after I got back and readjusted and was catching up, and they're like, hey, why haven't you run my shed? So that's one reason why I feel bad that that I let.
That's fair.
The ones that got given to Sean in Texas, I think they did run those real fast for everybody. But yeah, I'll have to make sure if I collect, or when I collect sheds at shows again, I got to look at them like at the airport when I'm on the way home and make sure I put the paid ones to the top and get those run right away. But yeah, we'll look into that. Cal the Blood says. On Instagram says, I'd love to see a diagram comparison between all the genetics and the yellow belly complex. So we've talked about those. I think, did we talk about the yellow belly complex all in one episode, or did we split those up? We split them up.
I think we split it up, or rather, I think we have two different episodes. One for, I think maybe two or three. Like we split it up into like each sections of the yellow belly complex.
Yeah, I think we did like yellow belly, and then we did like asphalt and gravel together, and
yeah, that's it.
Something like that. Yeah, that's a good suggestion. We should do that. There is a good diagram in the paper that we could show, and I guess Kayla, if you have access to that paper fairly quick, we could even pull up that diagram.
I should be able to get that.
I can tell you what to type in if you want.
Oh, yes, please. Here. Yeah, what is it?
If you look up Hannah Seidel, Python Regis, and leucism.
Leucism and stripe formation in ball pythons. That sounds familiar. Yeah.
So I think there is a good diagram there, and any of you watching, you can just Google that same search, those same search terms.
Yeah.
But they do have. I know that if you're not used to reading scientific papers, it can be kind of overwhelming. There's all these words and all this weird stuff and these terms you don't know. But if you just kind of skim through it, there are things you can pull out of it. So yeah, you can kind of see the top part there where it shows like an ivory on the far left. And so they're talking about how the different types of mutations, the type of mutation that causes ivory, the -1 and the -17, those are both frame shifts that cause the gene to stop early. And so that's very disruptive and makes a big change. And that's why you get that ivory. And then the Spectre Spark is a little less intense of a change. And then gravel asphalt's even less. And then you get to the normal. I think that gravel and asphalt, if I remember right, those are changes in splicing. So that's how the different parts of the gene are put together. The sequence of that gene can actually be put in different ways. It's kind of like having 7 blocks that you know, Lego blocks, and you can make a tower with all 7 blocks, or you can make it with 5. So it's still a tower, it's just a little different, and that's kind of like a splice difference. You can make 2 different proteins with the same gene and then just splice it or use different parts of it a little differently. And you can you can have one coding sequence, but you can actually make 2 different proteins that are slightly different. And if I remember right, gravel and asphalt were splice variants. And then Spark and Spectre there, I would imagine those are missense mutations. I don't know if it says it right there, but yeah, that's just essentially changing one amino acid to another. So yeah, it's a pretty cool paper, and they've got, well, not just Dr. Seidel, but the students that worked with her and other researchers. They did a great job. So this Fell the Blood saying it's just weird that the RK line asphalt stuff I have is so much more reactive than typical yellow belly complex stuff. So far, I think it's the best yellow belly complex gene to work with. Yeah, and I know you've sent us RK asphalt, Cow the Blood. If you know other people that do, tell them to send them too. We could use a handful more. That would help out. But yeah, once we've located it and know for sure where it's at, then we can kind of see if what kind of different mutation it is, you know.
Yeah.
If it's also a splice variant like asphalt is, or if it's a little different, that would be interesting to know.
Mm-hmm.
But we're not seeing it like right near where asphalt is. Rec Room Snake says hi. How's it going? I was just telling everyone, Mary, that's Mary. I was just telling everyone I felt bad that they're saying we should partner up with shows and do a discount and get sheds turned in at shows and give people a better deal. And I said I made the mistake of getting sheds at shows and not realizing some of them were paid, and some of those were Mary's. Sorry, Mary.
Oh, Mary.
So when I collect sheds at shows, I will do a better job. I'll think of Mary and her awesome little, you have to tell me, Mary, in the comments the name of your little dog I met at the last show I saw you and your husband. That was a pretty sweet little dog. But yeah, anyway, I will make sure that I do a good job of at the airport on the way home. I'll put all the paid ones at the top and know that I need to get them run for you.
There you go. Let's see. Did you talk about Prophecy? Hey, good to see you again.
Mary says, "Nah, you're good. You're very kind." But what's the name of your dog? We gotta.
Oh, yeah.
You have an awesome dog. So if they see you, Binks. Okay. So any of you that see Rec Room Snakes, they do lots of shows on the East Coast. Binks.
Binks. That's an adorable name.
Adorable dog too.
Nice. It's a good dog.
Yeah. Coiled Up Snake says, "What's up, RGI squad? Good
Hello.
to see you, Coiled Up." And I was just saying earlier, I will be going at least to the Pomona show. I don't know if I'll make it to any other California shows. I think you're more Northern California. You have to let us know if you come down to any of the Southern California shows.
Yeah. Here's a couple topics back. Here's the I think the thing you were referring to, talking about like what type of variations these different ones were.
Yeah. So that first one, so those are frame shifts, a -1 or a -17. So those of you that had like high school biology, you have the DNA genetic code. Every 3 A, T, C, G, each of those 3, that's called a codon. And so in the language of changing DNA into RNA and then into protein, each word is 3 DNA bases or 3 nucleotides. And so like ATG is the start codon. That's the first little bit of sequence that says start making this protein here. So any change that's not a multiple of 3 will be a frame shift. So a -1, if every 3 is an amino acid, that's the code for an amino acid, and you take 1 away, that means every single codon after that's messed up because it's out of frame. That's why it's called a frame shift.
Okay.
So everything after that in the sequence is going to make the wrong amino acids in the protein, and then it will also likely stop codon sooner than it should be, and that's why it's so disruptive. And you just make a almost completely white snake. And then Spark and Spectre, those are missense. Yeah, so I was remembering right. Those are missense mutations. So you're changing. There's still the same number of nucleotides, so you're not messing up those sets of 3, but you're changing maybe a T to an A or a C to a T or whatever, and that's making it so the amino acid changes from one amino acid to another. That's why it's called a missense, because it's not the right amino acid for that protein. It's different. It's a different amino acid in there, and that can change how it settles in. Like those of you that have seen when we've talked about some of the other mutations, if it affects the part of the protein that's sticking out of the cell or inside of the membrane or inside the cell itself, any of those 3 places, if it changes the structure, it can alter how it interacts with other proteins around it, stuff like that. So that's why the missense is not as big of a change as the frame shift, but it's still a pretty big, can still be a pretty big change. And then the shift for the splicing, that's can be, fairly small. So yeah.
Fair enough. I really liked how you, Soda Ball said that too. I liked how you described frame shift. Like I had a small aha moment. Like I know you've explained it before, but for some reason, like everything's kind of moved over. Just like that, it made my brain click a little bit. So
yeah,
I appreciate that.
Yeah. Pictures help even more, but hopefully that was good. Morpheus Morphs BP on Instagram says, have you tested any sheds that had more than 2 yellow belly complex results in one shed? And the answer is no. That's extremely unlikely to happen, which that's probably what most of you realize and understand. That's not something that should happen very often. For that to happen, you would have to have 2 different animals breed, and when there's that crossing over happen, so say one is an ivory and the other one is a super spectre, so all the babies should be ivory and super spectre. But if you can get a crossover event where that baby should have been an ivory, but the crossing over happened between where spectre and yellow belly mutations are, you could actually have 3 different messages. You could have on one chromosome, you could have the yellow belly mutation. On the other chromosome, you would have yellow belly and you would have spectre. But it requires there to be a crossover event in that very extremely small spot. So a lot of the time, these chromosomes have tens of millions to 100 or 200 million base pairs on it. And the distance between yellow belly and spectre might be, I can't remember, it's in that paper, but say it's 10,000 base pairs. So for there to be a crossover event in that little tiny, when Dr. Travis Wyman was on and we were talking about this, he said it's like thinking about there's a massive earthquake and there's a big old fissure forms, and if it's you and your wife are sitting in the same house, it's not likely that big fissure is going to be between you and your wife. But if you're in, one end of the state and your wife's on the other end of the state, it's a lot more likely you're going to have that happen in between. That's the same thing. So it's not, we've never seen 3 different yellow belly complex mutations in one animal. It's extremely unlikely it would ever happen, but it's not impossible. So that is kind of a fun, nerdy question to think about.
Yeah. Let's see. We're just scrolling through that paper and mostly frying my brain, honestly. Okay, we're at the acknowledgements now. So
Yes. It sounds like Morpheus Morphs was talking to Heather Raffy about this as well. So yeah,
Oh,
fun
cool.
stuff can happen. It's not
Yeah.
likely, but it can, and it can make your brain hurt. Yes.
Cool. Let's see. Oh, actually, there were a couple of cool questions before I pull up those. Okay. Prophecy, exotics, and gaming. So there was a paper on how blackhead essentially turns spider into a wild type genetically. Now that you have both tests, are you seeing the same thing?
I'm not aware of that test, of that
Yeah.
paper, other than them saying that breeders, have observed that. If they gave any kind of mechanism for that, I would love to see that paper. So if you
Yeah.
know what it was, feel free to post either the title or a link to it. I would love to check it out. If all they
Yeah.
did was kind of mention in passing that breeders have seen that kind of cancels out, then we know that, and we see that. But as far as like any kind of a mechanism, that would be super interesting. That would be a cool paper.
Yeah. So I guess like maybe like it kind of turns spider into a wild type, maybe phenotypically, what we're seeing as breeders, maybe.
Yeah. When you look
Yeah.
at a spider blackhead, you can't tell really
But
that
it's either.
it just kind of
Interesting.
makes it look like a normal, and it doesn't have any of the neurological problems that you see with spiders. So
Huh. That's really cool.
yeah, it's very interesting.
Yeah. No, I'd love to see more about that. That's awesome. Also, hi Leviathan.
Hey.
We sponsor those guys. They're awesome.
Yes.
Um, let's see. Mike is asking, I know you use PCR gel for fast tests. Well, I know we use it for do we use PCR gel for fast tests as well, or are we just using those for sex determination?
Oh, we do different things. And so there's so many
Yeah.
different tests that we're doing, the types of
Mhm.
mutations like we were talking about earlier, if it's a -1, a -17, if it's a base change or a SNP. So it's hard to make everything be the exact same test. But as far as an at-home test in the future, so like if you think about like at-home COVID tests or like a pregnancy test, the reason those are able to get made is because there's millions being ordered. So if we all of a sudden had, which should happen, everybody should have reptiles in the complete the whole world. Even if it was just the United States, we have, all of a sudden, instead of having about 10 million homes or whatever it is now, 10 or 15 million homes, if we have 315 million homes that have reptiles, and we have a whole bunch of orders coming, then there'd be a lot of money to be able to develop tests like that are really simple and foolproof and can be done at home by untrained. Essentially, you'd be like an untrained technician. But to get to that point, there has to be a lot of research and development go into it. And it's just not feasible yet, but you all keep helping people see how awesome reptiles are, and more people do it maybe in the future. And obviously technology can improve and get better and faster and cheaper. So maybe 5 or 10 or 15 years, being able to develop more simple at-home tests will be possible. But yeah, right now I would say for you to be able to do any of this at home, you certainly would need some training, and you would go through some chunks of money, making mistakes and things like that, because anybody would. I did. The only reason I can do what I do is because I've made pretty much every mistake a person can make over the last almost 25 years. So yeah, that there would be a significant cost in getting set up, and then getting to where you can do it reliably without making mistakes. But it can be done. It's just not super simple. But we have some ideas of what we can do to be able to make it a little easier or a little cheaper for breeders that don't mind doing some of the test prep stuff. And that's something that we're kind of working towards making available. So those of you that want to do some of it on your own, we might be able to make that available in the future. So, but not going to be this month.
Fair enough. Let's see. Sloan Morphs is asking, do you know if the two different lines of asphalt are compatible?
I don't. So, Cat of
Oh,
Blood, if you're still on here, let us know if you've bred them. I think Leviathan, I think he had, Steven had some information or knowledge of how the RK asphalt interacted. My guess is, from what I've seen people say, they at least know the combos with RK asphalt, like Freeway or something like that, that those can look a little different, but like whether we know for sure that it's like allelic, that kind of stuff, I haven't seen that data. I don't know that myself. I think that people are assuming that because it looks so similar to asphalt. But yeah, if any of you that are listening know the answer to that for sure, that people have done the breeding and seen, it's like if you have an RK asphalt highway or freeway, I mean, and do that breeding, do you only get RK asphalts and yellow bellies? That would be a good indication that it is indeed in the same spot. Because you have to remember, like with axanthic, we have a very similar, hard to tell apart look that's on 3 completely different chromosomes. So it's possible that RK asphalt looks very similar to asphalt, but it's on a completely different chromosome. That's possible. It's not likely, but it is possible.
Cool. Prophecy updated their question. Canada, so I guess Canadian group, says that blackhead restores the spider's ability to develop pattern plus neurological system, which essentially makes it look normal, as spider itself causes a lack of pattern production plus neuro issues.
Yeah. I mean, that's certainly what we've seen as breeders. If they've published, then I've missed it, and I would love to see it. But yeah, if that's just what they're like saying in their posts and stuff, that definitely makes very good sense.
Oh yeah. Oh, Leviathan answered question previously. RK freeways look like super asphalts.
That's right. That's what they said before. But
Cool.
do you know, Stephen, or if those breed like freeways, like if you breed them, do you get all either RK asphalts or yellow bellies? That would be another question that we just assumed that you would, but we don't know for sure until we do the breeding or until we find it and know for sure if it's in the same gene or not. So if you happen to know the answer to that, I would also be interested in that.
Yeah.
And that's a good question, Matthew. I don't know. It says, if you do a blackhead spider pied, will they have pattern? I don't know. You would think it would equal it out, and it would look like kind of a normal pied, or maybe a little bit darker of a pied, maybe. I don't know if somebody's done that.
Yeah.
All right. So yeah, sounds like we don't know for sure from breeding yet. I would be very surprised if it wasn't in the same gene, since it looks so similar, and then it would act like a super, just like asphalt would. That's most likely what we'll see. But if anyone knows for sure from breeding, that would be interesting to see.
Mm-hmm.
So Matthew, you are working the blackhead spider pied. So yeah, that would be cool to see a picture of that.
Yeah.
And we can do a genetic test to make sure that it tests out that way too. That would be interesting to see, especially if it just looks completely like a normal pied. That would be interesting.
That'd be cool. Matt was also asking about green tree python sex tests.
So we have worked on that a lot. That's actually the original test that Sean and I, when we met, we were both breeding lots of carpet pythons, and we were very interested in the genus Morelia in general, and we knew how valuable a sex determination test for green tree pythons would be. So that's something that we have wanted from the very beginning. We've worked on it a lot. But
kind of our white whale, honestly.
Yes, or for the one I always say is it's our Eleanor, for those of you that are Seconds fans. So yeah, in recent, like in the last 6 months to a year, there's some research that is showing the sex chromosomes in the genus Morelia are different, significantly different from the sex chromosomes in all other python genera. So like Python genus, Aspidites, Lyaeus, all of that. It seems that the sex chromosomes in Morelia are different. And so that's probably why we've had such a difficult time. And it's not just us, other people have tried to do that as well, find a good sex determination test for green trees, and it's just tough to do. And it's possible that it's not even going to be something we can do that would be cost effective. It might be complicated enough to do that test that it would be expensive enough that it's not worth doing. Right now we have one that works for most sires, for the offspring of most sires. We can do it, but it costs like between $200-$300 a test. So it's expensive. And then every once in a while there is a sire that the markers we're looking at, that sire is homozygous for all those markers, and so he's giving them to his male offspring and his female offspring, so the test doesn't work. So it's not really very fair to offer it to the general public, because some people will send them in, and not only do we have to test the unknown offspring, we also have to have the shed from the sire and from the paternal grandsire, or from the sire and a known offspring from the sire that's a male offspring, because we have to match those markers on the sex chromosome that are going to the male offspring. And then we do that test, and we can say, okay, these animals have that, that marker or those markers that we know this sire gives to his male offspring. These ones don't. That's how we know males and females. And for the sires it works for, has worked perfectly so far. We did it probably 4 or 5 years back. We did it for 2 or 3 years and had perfect results. But it's so expensive and time consuming. And then, like I said, we had one breeder in particular. He had a sire that was very valuable, and he wanted to be able to determine the sex of the offspring. And that test for that sire, he was homozygous for all the markers we were looking at. And so unfortunately we couldn't tell him anything. So it's expensive, and it's kind of a letdown for a customer if you say, hey, we've spent, $300 or $400 or $500 testing out your sire and his markers, and we found out we can't determine the sex of his offspring at this time. So it's not a great test. So I think I'm still optimistic that we can get it eventually, but Morelia in particular, so that includes green trees and carpet pythons, that it's going to be a difficult group to get a sex determination test for, unfortunately.
We did talk a bit more about this about a year ago on our 36th episode of Reptile Genetics Weekly, back when I was still using like the old thumbnail format. So we do have that. That's back when we had the like that puzzle giveaway. That was cool. You were going to Pomona that week, or around there. But yeah, we did talk about that, and I just find that whole situation so interesting with green trees, because aren't Morelia like more of a like a more basal or primitive lineage of snakes, as far as we can tell? Or am I
no, not compared to Python, the genus Python. No.
Okay, gotcha. Never mind.
But I mean, that's according to phylogenetics right now, and over time, sometimes those change. So
yeah.
I believe
Anthropus in general.
Yeah. I believe the genus Python within the Python Pythonidae, I believe the genus Python is the most basal.
But yeah. And then there's that. I know boas are a little trickier too, whereas things for the genetic side, just like having solid sex chromosomes like ZW, is a lot more simple in vipers, elapids, and colubrids, which are as far as we can tell, a more a younger lineage. So at least I'm fairly sure, because I know those are kind of wrapped up in.
They are more derived.
More derived. Yeah.
Yes.
I found that so
really
interesting.
the reason that makes their sex chromosome so easy to work with is because, like us, we have a big X compared to a little tiny Y.
Yeah.
Well, in the advanced snakes or the Colubridae, the let's see, I think it's the Cenophidians is the group that includes venomous and the colubrids. But anyway, in that more advanced group, the Z and the W are very big. There's a big Z and a little tiny W. So when I'm trying to design a DNA test that can tell whether an animal has two big Z's or one big Z and one little W, it makes it a lot easier for me to design it so that it's, you know, a part that's only sequence on a Z or only sequence on a W. It's really easy to do that when there's a big sequence difference. But in pythons and boas, the X and the Y are both the same size. And even if we do like a chromosome spread and a staining, like some of you may have seen that, a karyotype, they still, with the banding, they still look the same. You can't tell them apart. You have to really go in and do a very fine look. And even then, for some species, that's difficult to do. We don't know where to look, where the difference is, because they're so similar.
Yeah, fair enough. So glad you like the this format, the soda balls. I've enjoyed it too. I think it's cool.
Yeah, and we've had, you know, a good 30, 40, 50 people on here. That's great. We appreciate it.
Especially for middle of the day on a work day for most people. So that's awesome. And we're actually like 4 minutes shy of an hour. So yeah, that's really cool.
Yes.
Awesome. Also, hi Smaze Serpents and stuff. Good to see you.
Yes, hello.
Let's see. Brian Young Peters asking if there are any updates on hognose testing, or any specific sheds we need from the community to help with research.
So for hognoses, still need all of them. You know, send all that you can. Probably what we'll get first will be the kind of similar mutations we see in other species, because we know where to look. So like albino, axanthic, hypo, lavender, those kinds of mutations will likely be what we'll pick up in hognoses. But if there's a ton of people that send in, you know, Arctic or sable or you know,
lavender
and conda or whatever, whatever we get a whole bunch in for, we will definitely sequence that. If we get, you know, 30 or 40 in of something, we know a lot of people want that, and we have a lot of information to work from, so that those will be ones that we'll definitely put work into. So now is the time. If you want hognose tests, you yourself send in a bunch and have people, you know, send them in, and the more we get, the faster we can do them.
Oh yeah. I know, well, actually I have a big box of hognose. I think granted, I know you've been getting a lot too, Ben. But I think when I was going through them and sorting those out, decent amount of albino, decent amount of, not a ton of leucistics. I know that's a big money ticket for leucistic hogs. Granted, we don't know what, like, there are a couple different places we could look to for like figuring out where leucism could be for hogs, but yeah, a decent amount of lavenders, lots and lots of condas, which that one would be kind of that one. I feel like that one would be less, like, lower priority, just because it's not like a recessive that's hiding. Usually you can tell visually, oh yeah, it's this. Not seeing a lot of caramels or any of the hypos, anything like that. So yeah, basically anything you want to send. All of it's good. All of it's helpful.
Yeah, we will take them.
Yes.
This is a question about calico. So for calico, we do have a decent amount of sheds, and we are working on that. So you definitely feel free, if you have calico or sugar animals, that would be great.
Yeah.
I'm in.
Can't wait to find out the results of Nightmare. Did you send in a fast test sometimes, like recently?
This is Matthew we did the episode with.
Oh, yeah.
Yeah, so that's one of the animals.
Yes. Yep.
Yeah.
Ooh. Oh, Leviathan with the good question. Oh my gosh. This might not be possible, but do you have data on the most mis-ID'd morphs or the most likely genes to pop up unexpectedly?
We don't, and I don't know how well received that would be. Some people would get frustrated if we did put up such a list. But that's something we could put some information together if people were not gonna have feelings hurt or anything. But yeah, it's also kind of hard. Like sometimes we'll get an unexpected thing and we can test the siren dam and we're like, oh, we figured it out, it's really easy. Or even from that one individual, and we can test it for other things. Like when Justin had an all white snake and he knew from the pairing that the I think actually maybe neither of them had a blue-eyed Lucy gene. But
Yeah, I
anyway,
remember
it
that one.
ended up that those two animals were from, it was a male and a female that were from the same clutch, but they were like triple head or quad head or something. And so they had been bred together to be able to get all those heads together. And it ended up getting white snakes. So we thought, okay, maybe one of them has a blue-eyed Lucy in it, and this is either parthenogenesis or androgenesis. And in the end, we tested the parents and tested them and found out that both of the animals had Russo in them. And so they were just super Russos, and it was completely normal sexual reproduction. So yeah, but there are times where we can't chase it down. Like maybe somebody purchased it and they don't have access to the siren dam or any other lineage information, and then we don't really know for sure. All we know is we got a weird result and we might think we know what happened, but if we can't chase it down, then we don't really know for sure. So yeah. Oh, and you did say there would be feelings hurt.
Yeah.
Yeah, I would imagine. What was the next one? I hadn't read that yet.
Oh, no, that was it. I just put the original question back
Going
up.
back to that.
Yeah, no, I like it though. Yeah. Well, we're at about an hour now. So do we want to? Do we have any other last questions on Instagram or anything like that?
Yeah, if anyone has any last minute questions, we'll go another minute or two, and then we'll call it for today. I don't see it coming through yet. Oh, I guess Duskfire Geckos did ask, is the spider morph and its neuro issue by chance related to PMEL or PMEL 17 specifically? So since it's, to my knowledge, not published yet, definitely let me know if I missed it. I'm not going to confirm or deny too much about specific genes that are related or not related, but I think that whether it's us or the Canadian group or someone else, I think that will get published here before too long, and you'll be able to have a very, very specific story that's a cool story.
Yeah. Mike added on to the Leviathan's question. I feel like the older recessive pied, hypo, albino would be floating around everywhere. Found a lot of hypo with your testing. Yeah, checks out.
Yeah, we've definitely found albino, clown, hypo, pied, yellow belly, inchy, ultramel, even candy. There's been two or three, not lots, but there's been two or three that ended up having candy that they didn't know it was albino or candy in the pairing or from the person they got it from, but there's definite candy there as a het. So yeah, those all show up. And Leviathan had one in Sunset, ended up being an OD Sunset. So that was pretty cool. If I remember right, Stephen or Courtney, whichever of you, I think someone said it was Stephen, right? But whichever of you is on, have to remind me if that was a panel test. I think it was a panel test and was just completely unexpected. But yeah, so now they know they've got OD in one of their Sunset lineages. So
that's cool.
Yes.
I guess before we call it a day, if there aren't any more questions, I'm gonna drop this banger of a post that Ben screenshotted from Inky Clouds. It's beautiful. I cackled when Ben sent it to me.
Yeah, and the other beautiful thing about that is, some people that might pop up and just because it's a chart, they don't even look at it at all. But even if you just look at it briefly, you're like, okay, those colors don't match up, and you're like, okay, I'll read this. Like,
Yeah.
yeah, and then someone in the comments said it should be all one color, and that one color should be next year is going to be my year.
Yeah, sounds right.
Yeah.
We'll get that clutch next year, guys.
Funny stuff. Well, thanks for being a part of this with us. And if you're at work and you didn't get busted, then good job. If you're at work and you did get busted, send us a sad story and maybe we'll hook you up.
Yeah. Or if you did get busted, then we'll get good.
Yes.
Or Ben's was nicer. Mine, yeah. But yeah.
You had a video. You had a video to show, Kayla. You got to show the video.
Oh yeah. Oh my gosh. Okay. Okay. Last thing. Then
we'll really go.
Okay. Yeah. Where did I hide it? It's my boy, Mr. Lemon Bars.
Yeah, I got some cool breeding activity going on, and many of us are probably ball python breeders, but this was something I hadn't seen anything quite like this. In boas, I had a litter of common boas. I would say I kind of saw something similar with them, but those of you out there that have done other colubrids, maybe you've seen this.
Yeah, no, so for context, I paired them he's still covered in shed and she's kind of in blue. The only reason I paired them so early after brumation while they still had some shed going on was because the males were coming out of brumation. I put them together while they're still cold and not going to fight. And then out of nowhere, like one of the males decided, okay, it's time to start like wrestling with the other male. I'm like, okay, I assume the females must be putting out hormones and stuff now. So I just pulled the boys apart, threw them in with their girlfriends early, and just immediately we got breeding activity. So this is Mr. Lemon Bars putting the moves on Clover. And I love this little undulation he does. It is the cutest thing ever. And it's just so interesting. I don't know of other species that do like the little body rolls or not, but it's like just look at him go.
Massage going on.
Like how cute. And she does kind of like a little tail waggle thing, but yeah, like probably like 3 minutes after this, they locked up. And it was the very first time she had locked up. I tried to get them last year, but to do it last year, she was having none of it. But this year 4, we're good.
Cool.
Yeah.
That's exciting.
I know. I'm hoping for double hets this year from those guys. So fingers crossed.
Awesome.
Yeah.
And Leviathan's original. So the very first one you got. That's awesome. That's a good way to start.
Yeah, it is. Oh, that's awesome.
All right. Well, my kids are home for spring break this week, so I got to go so I don't get in trouble. I got to go help out.
There. All right. Awesome. I'm going to hop off here and go do a grocery run, maybe hit the gym. So
yes.
Thanks, guys, for joining us.
Thanks for joining.
Yeah.
See y'all Tuesday night for the recorded show, and next Thursday we'll do this again. We definitely appreciate it. Lots of interaction, and so we'll keep it coming.
Awesome. See you guys.